All right, we are on to evolution days 9 through 11. Uh, just gonna kind of jump into it. The first one that we're gonna talk about is a few definitions that have to do with phylogenetic trees. So with phylogenetic trees, I'll draw one out like this. We start right here, the root. That's what everything branches off of. Okay. Everything branches off of the root. Um, and then our first branch out point is called the basal taxon. Basal taxon. Or sorry, our next one that we're going to go over is called a sister taxa. Sister taxa is when we have two branch out points coming off of one branch out point. And the last one we have is a polytomy. Okay, a polytomy, poly meaning multiple, is just when we have multiple polytomies coming out. We should also note that anytime, anywhere on this whole entire thing, we have a place where they branch out. This can be called a node, otherwise known as a branch point. Okay. You guys have already done quite a bit of practice with this in your homework, hopefully by this point. So I'm not going to really go over how to do it. And so, yeah, really the only other thing that we're going to talk about in this is number one, how can we name different stuff, and how can we classify different stuff? So first we'll start with classification. Carlos Linnaeus, I think that sounds right. Carl Linnaeus, sorry, not Carlos. Carl Linnaeus came up with this classification system. And the one we don't really worry about the top one, which is life. So we just worry about uh domain, kingdom. All right, now I guess domain, kingdom, phylum, class, order, family, genus, and lastly, species. Fuck yes. Okay, and the way that I learned about this is a mnemonic. You just need to be able to memorize it, basically. But the mnemonic I learned is dumb, kids, playing, chess, on, freeway, get smashed. Pretty simple. DKPCOFGS. However you need to memorize that, just memorize that. What you should know is the way they ask these questions oftentimes is going to be like a genus is made up of a bunch of, and the answer would be a bunch of species, right? So a bunch of species makes up a genus. A bunch of genus makes up a family, right? A bunch of families makes up an order, and so on. Or they could ask you, like they could say something, a bunch of classes makes up and the answer to that would be a phylum, right? So a bunch of classes makes up a phylum. It's just the opposite way of asking basically the same question. So one of them will be asking you what something is made up of, and the other one they'll be telling you what it's made of and asking you what it is. And just being able to answer that is pretty important, and being able to figure out which one they are. It's multiple choice, so you don't like none of these letters are the same letter. So as long as you can remember the letters, you should be able to figure out which ones go in which order. From there, we'll talk about how we can name these. And whenever we name, we do it just with the genus first and then the species. You practice this in the lab if you pick the lab. but it can be genus and then species. And it needs to be like this, capital on the first one, lowercase on the second one. Okay. Uh, Something else to notice is that the second name is in italics. And the first name is regular. So I can't write in italics. That's part of the reason why this doesn't work super well. But I can try a little bit, right? So it would be genus and then species. Another way to write this is we can abbreviate the first one, and then we still have to italicize the second one, right? And we still have to capitalize the first one. These are the two possible ways that we can write it. But any other way is invalid. So you'll see that potentially on your exam, and it will give you a bunch of different ways. If it's not written like this or like this, and obviously replaced with real genus names or real species names, or they could even do it like just how I did it right here, then it would be invalid. So just make sure you know that. Other than that, that is all of day 9. Day 9 is a fairly simple, fast day, because you're kind of just getting introduced to phylogenetics. I guess we could go over a few definitions briefly. A taxon is just a group of organisms, specifically a group of organisms at any taxonomic level, but really just a group of organisms. Okay. Phylogeny is the evolutionary history of a taxon. And other than that, the word nomenclature just means naming. So in this case, it's the scientific process by which we name things, or the orderly application of names to taxonomic ranks. But if you can just remember nomenclature, naming, good enough. Really good enough. And I think that is it for day 9. So on to day 10. Just pull this up really quickly. So for day 10, we are going to basically do something involving cladistics. Now, cladistics is just a way to group things. A clade is a group. Okay, that's all you need to understand for that. Clade is a group. Cladistics is how we group things. We can group things in lots of different ways, and that's what we're going to talk about really quickly. So we'll draw out another tree, and it looks like this. And I need to grab a few other colors just really quickly. So that I can label these. And the first one we're going to label, first type of clade that we can have is called a monophyly. Okay. So a monophyly or monophilic. This is basically just saying that we have all descendants from one common ancestor. Okay, so if I drew a line, what I like to do is if I just look at the circle, right? It's kind of hard to see on this camera. The orange, isn't it? But you can see I have my circle. And if I just draw a line right outside that circle, right where it began, earliest point, and I can see that I have every single descendant after that within that circle. And that makes that a monophyly. Now use better colors on this for the next ones. That way you can hopefully see a little bit better. So the next one we're going to do is called a paraphyly. And I'll do that one in blue. So a paraphyly would be something like this. And a paraphyly or paraphilic is some, but not all, descendants from one ancestor. So you'll notice that right here I drew the line for this other one. That was my ancestor. And then I had every single one after that, right? But here if I draw my line, and I'll even erase this and make it come around like that. If I draw my line right there, right? I have all of them so far. This one, this descendant, this descendant, this descendant. But if I keep going down my descendants, because these are all descendants of this ancestor, I don't have these within my circle, within my clade is probably a better way of saying that. And therefore that's a paraphyly because I have some, but not all of my descendants, right? And so if I circled these two right here, that would be a paraphyly, right? If I circled all of them, that would be a monophyly because all of them come from a single descendant. That's where the root is, if that makes sense. So you can kind of see how we do that. There's lots of different ways to create a clade. The last one we're going to talk about is called a polyphyly. And a polyphyly, we just represent like right here. We could also represent a polyphyly. That would be a paraphyly actually. But you can see if we went out, right? And we go right outside of this and we show the two different ancestors that we have we have a polyphyly, and that's exactly what a polyphyly is, or paraphilic, right? That is multiple descendants from more than one ancestor. Okay. Hopefully that makes a little bit of sense. I'm sorry that it's not super clear on the camera, but it's also on the slides. And hopefully those definitions kind of help with clades. So the next thing we're going to talk about, I believe, is going to be parsimony. So parsimony is just the idea that we want the simplest possible phylogenetic tree. The simplest possible tree. If I drew out, let's see if I can do this. A tree like this, right? And it just went like that, right? Versus if I drew out a tree, right? So here we have 1, 2, 3, 4 descendants, okay? And now if I drew out a tree, one, two, three, 1, 2, 3, 4. Okay. So here, right, I have points of difference. We're talking about points of difference. Here I have 1 node, 2 nodes, 3 nodes, right? Whereas here I have 1 node, 2 node, 3 node. Those would have equal parsimony, okay? But right here, and this would take me a little while to draw out, so I'm not really going to draw it out. But on the slides, right about here. Okay, I have two different phylogenetic trees. Okay. In this one, I have 5 points of difference. In this one, I have 1, 2, 3, 4, 5 points of difference. But you can see that this one's way simpler and way quicker, whereas this one has a bunch of different, like, okay, we're splitting here, we're putting two splits on this one, we're putting one split on this one. This one just has one split off of each one. We always want to just have one split off of each one if possible, even though we can do two splits. And so I said before that these have the same parsimony because they have the same number of splits. That's actually incorrect. This one has better parsimony because it has only one split. It has only one split per node. Whereas this one has two splits off of one node. And so we want the one with one split off of each node every time. That would be the simplest way. Other than that, a lot of phylogenetics. We do need to talk about horizontal gene transfer. We'll mention that briefly. Horizontal gene transfer is basically when we take a phylogenetic tree, so, right, usually descendants just go down, right, go down, like that, right? And you could think about like your family tree, okay? Right, you got all of your stuff. That would be a vertical, right, gene transfer. You get all of your stuff from your parents, okay? Well, and in a horizontal gene transfer, you could have like your grandma who's given you DNA, okay? And this will happen a lot of times in prokaryotes. So we'll talk specifically about prokaryotes first. In prokaryotes, you already learned about this with bacteria specifically. And so they do this in three different ways. They do it through transduction. And you don't need to know it as in depth for 181 as you do for 180. But you have transduction, transformation, and you will have conjugation, right? And all we're doing is we're taking DNA that wasn't given from the parents and we're putting it into these bacteria or these prokaryotes in some way. So our first way with transduction is through a virus, right? Our second way with transformation is through naked DNA, right? DNA, they just stumbled upon it and then they absorb it. Our third one is going to be through conjugation, which is basically when the bacteria puts its pili in and transfers bacteria that way. So bacteria, pili. And those are the three different ways that we talk about for prokaryotes. Now, with eukaryotes, we'll briefly mention this. With eukaryotes, we have something called jumping genes, otherwise known as transposons. So transposons, I should say transposons. Transposons, otherwise known as jumping genes. I don't really know how this works exactly. And if you wanted to learn more about this, take genetics class. But basically what ends up happening is certain eukaryotes have the ability to absorb DNA matter somehow of other things that they come into contact with. A really good example, and I'm just going to kind of give examples because that's more what you're going to be tested on. Aphids do this with something called HGT. So green aphids, they eat this HGT and they turn red. And it's literally because their genetic code changes when they eat this stuff. Another example we have is corn. Corn does this, and I don't even know how it does it. To be honest, don't care. Just know that, and you should do well for your exam. Last thing we will talk about, I think, is the endosymbiont theory. And so we have three different theories. Of endosymbiont theory. I'll write it in different color, just the red's not that easy to see. Purple seems to do well. So when you hear the word symbiosis or any type of symbiont, I can show you what that sideways, whatever. Any type of any symbiont, what you're thinking is two animals or two beings that work together. Endo meaning inside, so one being working inside the other, and symbiote meaning, in this case, that they're both benefiting off of each other. So mitochondria in our cells is what we're talking about. Or you can also have it with cyanobacterium, and the cyanobacterium would do this in a lot of plants and stuff. And so with that, we have a few different ideas. We have number one, nucleus first. Number two, mitochondrion first. And number three, eukaryote first. Okay. And with that, these three hypotheses, I'll kind of show what they are. But with the nucleus first hypothesis, we get the idea that we take the nucleus and it goes inside the cell first. Okay. And then, so that happens first. And then second, we get our mitochondria coming in, right? So first the nucleus, then the mitochondria. Mitochondria first is the opposite, right? So it's basically just the same thing, and I'll just erase this, but it would go the nucleus second and the mitochondria first. Okay. The eukaryote idea is that the nucleus was always a part of it. Okay. So the idea for the eukaryote is that, right, we don't really care which one's first because there's only one thing going in. The nucleus was already in there, and then the mitochondria just comes in afterwards. And that's the idea of eukaryote hypothesis. So those are the three different kinds. And then we will talk about the different models. I guess I lied. I said that's the last thing, but I got one more thing to talk about. So we have three different models to represent different things. They explain different things. The first one is just the tree of life model. So the tree of life, otherwise known as the vertical gene transfer, vertical, with an A, vertical. And that's your most common, right? That's what you see on family tree, right? Fairly simple. Okay. The next one we have is the web of life. And this one's very similar to the tree of life, except for it allows us to represent now horizontal gene transfer. So if I draw it out like that, right, we could have gene transfer from here to here. And so you're seeing a little bit more complexity in that. And that would be for horizontal gene transfer. Okay. The last one that we're going to talk about that I'm going to show is going to be the ring of life model. Okay. And the ring of life model, we have archaea, we have eukaryotes, and we have prokaryotes. This one, I'll put real life. This one is the best explanation for the origin of life. And you should know that, okay? So, fairly simple, those three models. If you just know those three and know those things about them, then you're doing really well. Let me just make sure I didn't miss anything on day 10. Yeah, that should be good. Okay, on to day 11. Almost done. And forever. So, day um day 11, we're going to talk about a few things. We're kind of going to jump around a little bit, but the way that I've memorized this one is I just say 4, 4, 3, 2, it goes 4, 4, 3, 2, 2, 2, 1. Okay. And then I just kind of go throughout the whole thing. So, let me just make sure I get this right. I might need to pull this up briefly. I think I have it pulled up. Yep. So, one thing I'm missing. Okay, yeah. So about 4.6 billion years ago, we formed the Earth. Okay. 4.5 billion years ago, we formed the moon. Fairly simple. And then about 3 point, I think it's 3.5 billion years ago, but I just want to make sure I get the numbers right. Yeah. 3.5 billion years ago, I formed cyanobacteria. Sorry. I formed protocells. And protocells are basically the first prokaryotes. Okay. And then about 2.7 billion years ago, 2.7 to 2.3 billion years ago, I had the oxygen revolution. So oxygen revolution, and we'll come back to that. And then after that, about 2.1 billion years ago, I have the 2.1 billion years ago, I have the endosymbiosis. And then the last thing we have multicellularity. That happens about 1.5 billion years ago. 1.5 billion years ago. Okay. Now, with this, what do you need to know? Well, if you just know all this, you're doing pretty good. We do need to expand a little bit off of the oxygen revolution. So off the oxygen revolution, we have a few different points. Number one, rust forms, right? And that's due to oxidation or redox reactions. Okay, you probably learned about those in chem if you've taken any chem. We also have, we have the extinction of anaerobic. Anaerobic means that they don't use oxygen. So that kind of makes sense, right? When we start getting a bunch of oxygen, we have the extinction of the people who don't need oxygen. And so we're bringing in, I should probably write this right here, we're bringing in the aerobic, right? And with that, we also brought in cyanobacteria. And cyanobacteria, they do photosynthesis. And that's why we're producing oxygen at this point, right? Because they take carbon dioxide and they pump it out and make oxygen. Let me just make sure I'm not missing anything else. Yeah. Iron rust, cyanobacteria, photosynthesis, oxygen accumulation in the atmosphere, anaerobic prokaryotes. And then we have cell respiration that's going to start happening. So cell respiration, that's what we do. That's our type of metabolism, right? That's where we take oxygen and we form it into energy. You're doing that right now as you breathe. Other than that, I think we're doing good. We already talked about endosymbiosis and what that was. And then multicellularity, we just have the first fossil found from then. But other than that, we're just going to talk about a few different ideas about how we could even come up with this timeline using a few different scientific experiments. So those scientific experiments include number one, Miller-Urey. And so I'm trying to just pull this up. So Miller-Urey, they specifically focused on taking things from abiotic material to biotic material. And with that, they used lightning as their base theory for this to happen. Basically, they're taking things from not life, right, or inorganic to organic is another way of saying that. So inorganic to organic. And the next thing the next study that we're going to talk about is going to be the operant Haldane study. So the operant Haldane study, operant Haldane study, basically what they did, right, is they said, okay, we can take something from no life to life. That's what Millie Ray did. And operant Haldane came in and said, well, we need to not only take it from no life to life, but we need to be able to take it from unicellular to multicellular. And so they did that, and we call that monomers or unicellular. And we took that to polymers. And this needed a lot more energy. So the lead behind it is that volcanoes erupted, allowing for this to happen. Okay? And basically that's like saying prokaryotes to eukaryotes. If that makes sense, more sense in your head. With that, I think that we are getting pretty close. Yeah, that was the one I was missing. Okay. Yeah, we're getting pretty close to being done. We're going to talk about one more thing, and that is the fact that RNA came before DNA. And the reason why we think that is because RNA can do stuff that DNA can't. It can do everything DNA can, but it can do more, right? So RNA has a multi-purpose. It can hold genetic code just like DNA, but it can also lead to transcription and translation. It can serve multi-purpose. It can form ribosomes and proteins and stuff and kind of combine with all this other stuff. And so it is multi-purpose. And so we think that we need to have both of those things in order to actually produce life. And that's why DNA is not a great model, and RNA probably came before DNA. And those are all just kind of theories. Most of this is just theories. Other than that, I'm trying to see if there's anything else that we need to talk about. Yep, I think we're good. So good luck to you guys on both of your exams, your final and evolution, and I hope that these videos help. If you have any questions, you can shoot me a text. Hopefully I can answer as soon as possible. But thank you guys. Have a good one.